Welcome to Creative Diagnostics channel. This video we are going to talk about the use of viral vectors in vaccine development. Viral vectors offer several advantages compared to traditional subunit vaccines. One key benefit is their ability to elicit not only robust antibody responses but also cellular immune responses, which are crucial for eliminating pathogen-infected cells. Additionally, viral vectors can induce high immunogenicity without requiring adjuvants, while also generating durable immune protection, in some cases achieving effectiveness with just a single dose. Viral vectors can be engineered to deliver vaccine antigens to specific cells or tissues. To enhance their safety and reduce reactogenicity, many viral vectors have been genetically modified to become replication deficient. The antigens expressed by these vector-transduced cells induce immune responses, with the durability of antigen expression often correlating with sustained antibody and T-cell responses. Furthermore, the presence of antigens on the vector particle surface or within the viral structure also contributes to immune response induction. Although replication-deficient viral vectors are generally safer than replication-competent ones, they may require higher doses or prime boost regimens to elicit sufficient immunity, and depending on specific applications, replication-competent vectors might still remain the preferred choice. A key advantage of replication-competent vectors lies in their ability to mimic natural infection, triggering cytokine induction and co-stimulatory molecule activation that provides effective adjuvant effects. When replication-deficient viral vectors fail to stimulate optimal immune responses, the incorporation of adjuvants may become necessary to enhance immunity against the encoded antigens. Viral vector vaccines can be further optimized to improve transgene expression in target cells, enable dual antigen delivery, circumvent pre-existing immunity from repeated immunization, and mitigate potential side effects. These vaccines also offer flexible administration routes such as intranasal, oral, intradermal, or aerosol delivery. Such alternative immunization strategies can induce localized immune responses at mucosal sites, providing protection against or limiting respiratory and gastrointestinal infections. Viral vectors have been utilized in both preclinical and clinical trials as vaccines against various infectious diseases including HIV, malaria, Ebola, and more recently SARS-CoV-2. While offering distinct advantages in immunogenicity and safety profiles, they also carry associated risks that require careful evaluation. Current research explores diverse viral vector platforms such as adenovirus, poxvirus, herpes virus, vesicular stomatitis virus (VSV), and lentivirus in preclinical and clinical studies. These vectors are engineered to deliver vaccine payloads, stimulating robust immune responses against proteins produced by their encoded transgenes. Adenoviruses are a diverse family of DNA viruses capable of infecting respiratory, ocular, and gastrointestinal epithelial tissues in hosts, with linear double-stranded genomes ranging from 26 to 45 kb. Several adenoviral vectors serve as vaccine delivery systems due to advantages such as relatively low pathogenicity, genetic safety, and lack of integration into host genomes. Their strong immunogenicity, broad cellular tropism, and high-capacity transgene incorporation further enhance their utility. Adenoviral vectors are classified as replication-competent or replication-deficient based on the presence or absence of the E1 region. Adenovirus serotype 5 has been widely adopted as a viral vector platform in vaccine research due to its capacity to induce both humoral and cell-mediated immune responses. However, its application faces challenges from pre-existing immunity, with seroprevalence reaching up to 90% in some populations, which may impair vaccine-induced immunity. This limitation became evident in the STEP HIV vaccine trial, where Ad5 vectors expressing HIV-1 GAG, POL, and NEF genes were paradoxically associated with increased HIV susceptibility, a risk that escalated with higher baseline Ad5 neutralizing antibody titers. Despite these concerns, Ad5 vectors have been utilized in multiple SARS-CoV-2 vaccine studies. Preliminary immunogenicity data from a randomized, double-blind, placebo-controlled phase 2 trial demonstrated that a single dose of replication-deficient Ad5 vectors expressing SARS-CoV-2 spike protein elicited robust antibody responses and cytotoxic T-cell activation while maintaining an acceptable safety profile. The challenge of pre-existing immunity against Ad5 has driven the development of novel adenoviral vectors based on less prevalent human or animal-derived serotypes, notably chimpanzee adenoviruses, ChAd. Adenoviral vectors derived from ChAd serotypes have been engineered and tested against various pathogens including rabies, MERS and more recently SARS-CoV-2. Among these, the ChAdOx1 nCoV-19 vaccine targeting SARS-CoV-2 gained rapid momentum, advancing to Phase 3 clinical trials by 2020. In a Phase 1 of 2 trial, the replication-deficient ChAd vector expressing the SARS-CoV-2 spike protein was administered via intramuscular deltoid injection to 543 participants as a single dose, with a smaller subset, 10 individuals, receiving a booster at day 28. Neutralizing antibodies were detected in 91% of participants by day 28, rising to 100% seroconversion post-booster. Subsequent Phase 2 of 3 trials confirmed the vector's safety and immunogenicity across broader age groups. AAV vectors are the most popular choice for gene therapy and delivery of therapeutic antibodies thanks to their relatively low immunogenicity, high safety profile, broad tropism, and their tendency to maintain long-term gene expression. AAV vectors are produced through transfection of human embryonic kidney, HEK, 293 T cells with transgene, packaging and helper plasmids. Vector particles can be purified by polyethylene glycol (PEG) based precipitation, P-mediated protein removal and affinity chromatography. In recent years, their use as vaccine vectors has expanded for treating and preventing infectious diseases such as HIV, human papillomavirus and influenza. Multiple studies highlight that AAV vector vaccines can induce strong and durable antibody responses with just a single dose, eliminating the need for adjuvants. In some cases, they have demonstrated superior or longer-lasting antibody responses compared to other vaccination strategies, including DNA vaccines, recombinant proteins, inactivated viruses, or virus-like particles (VLPs). However, AAV vectors are considered less immunogenic relative to other viral vectors. The isolation of multiple AAV serotypes and capsid variants enables the development of prime/boost strategies, where AAV capsids can be switched to avoid neutralizing antibody responses against the capsid induced by initial priming. Key limitations of AAV vectors include their limited transgene capacity and widespread pre-existing immunity in humans. Strategies to enhance AAV immunogenicity and circumvent pre-existing immunity are currently under evaluation. Recombinant AAV vectors are particularly favored for passive immunization, such as delivering monoclonal antibodies and immunoglobulin derivatives, due to their ability to sustain long-term expression and high therapeutic protein levels. Vesicular stomatitis virus (VSV), a member of the Rhabdoviridae family, is a single-stranded negative-sense RNA virus characterized by its bullet-shaped morphology. VSV typically infects cattle, horses, pigs and goats, causing lesions on the gums, lips and tongue, sometimes accompanied by fever and excessive salivation. While transmission to humans is possible, infections are most often asymptomatic. The non-segmented viral genome encodes 5 proteins: N-nucleoprotein, P-phosphoprotein, M-matrix protein, G-glycoprotein, and L-large polymerase. The G protein or glycoprotein mediates viral particle attachment to host cells. VSV represents a valuable vaccine platform thanks to its ability to replicate at high titers, its low seroprevalence and little pre-existing immunity in humans. Recombinant VSV-based vectors, RVSV, have demonstrated safety and immunogenicity in preclinical and clinical trials, with the VSV-based Ebola vaccine, RVSV-ZBOV, recently approved for human use. The RVSV-ZBOV vaccine lacks the VSV-G gene, which enables wild-type virus to infect diverse cell types, and instead expresses the Ebola glycoprotein, GP. Clinical trials for this vaccine concluded by late 2018, proving its high efficacy across diverse populations. Both preclinical and clinical studies revealed elevated neutralizing IgM antibody levels, suggesting this antibody isotype may play a significant role in vaccine-induced immunity against EBOV. The RVSV platform is currently being evaluated for vaccines targeting other filoviruses, including Sudan virus, SUDV, and Bundibugyo virus, BDBV. as well as coronaviruses, Zika virus and Crimean Congo hemorrhagic fever, all showing encouraging results in preliminary research. Integrase-deficient lentiviral vectors, IDLVs, represent a promising vaccine platform due to their ability to induce potent and highly durable immune responses even after single-dose immunization. Derived from HIV or SIV through splitting the viral genome into separate plasmids, IDLVs are engineered to be replication defective and non-integrating by introducing mutations in the long terminal repeats, LTRs, packaging signals and integrase gene. These vectors persist as episomal forms in target cells, enabling sustained production of encoded proteins throughout the cell's lifespan. The non-integrating phenotype serves as a critical safety feature, mitigating the risk of insertional mutagenesis inherent in integrating viral vectors. IDLV is an attractive delivery platform due to little pre-existing immunity in humans that can be coupled with a vesicular stomatitis virus G-envelope glycoprotein VSV.G serotype exchange strategy to reduce antivector immunity for repeated IDLV injections. Following sequential vaccinations of rhesus monkeys with an IDLV expressing a series of HIV-1 envelope proteins, immune responses were strongly boosted after each of the 6 vaccinations performed over 3 years with no evidence of tolerance induction. In addition to being highly immunogenic and inducing durable responses, IDLV also demonstrated an excellent safety profile. Long-term immunity induced by IDLV may be ascribed to the persistence of IDLV in vivo. Other studies have also shown the efficacy of IDLV as a therapeutic vaccine as opposed to strictly prophylactic. Poxvirus-based vaccines have been used against various infectious diseases, such as HIV-1, tuberculosis and malaria. Poxviruses possess double-stranded DNA genomes capable of cytoplasmic replication and transcription using viral polymerases. Poxvirus-based vaccines have been employed against various infectious diseases, including HIV-1, tuberculosis, and malaria. One advantage of using poxviral vectors is their cargo capacity for heterologous genes of around 25 kb, which is far larger than that of other vectors. This makes poxviral vectors ideal candidates for the generation of multi-antigen vaccines against different pathogens. Poxvirus vectors have shown to be highly immunogenic and able to induce robust immune responses. The modified Vaccinia Ankara MVA vector shows promise as a vaccine delivery platform in poxvirus-based immunization strategies. A study on the MVA-vectored HIV-1 mosaic bivalent vaccine demonstrated its ability to induce both humoral and cell-mediated immune responses in humans, while maintaining favorable safety, tolerability, and immunogenicity profiles. Another replication-deficient viral strain used in vaccine research is the New York Vaccinia virus, NIVAC. Research indicates that NIVAC recombinants containing HIV GAG-PAL-NEF antigens and viral envelope genes can enhance both CD4 and CD8 T-cell responses. The same study revealed that combinations of MVA/NIVAC vectors produce broad-spectrum immune responses, while DNA/NIVAC combinations generate more robust immunological reactions. These findings are further supported by research comparing DNA vectors, NIVAC vectors and their combination, which showed that combined vector approaches elicit earlier and more potent antibody responses. Heterologous viral vector vaccines, HVVV, consist of a combination of 2 or more vectors encoding either the same or different antigens that can be administered together or in prime/boost regimens. HVVV immunizations can be advantageous to induce higher magnitude and more durable immune responses compared to homologous vector vaccination. Combination immunization of rhesus macaques with VSV Vaccinia virus (VV) and Ad5 vectors expressing SIV-MAC239 GAG together with 3M052 adjuvant protein resulted in 67% protection against viral challenge in vaccinated animals, which outperformed the 53% protection observed in animals receiving adjuvant protein alone. Notably, the study revealed that HVV vaccination induced potent GAG-specific CD8 T-cell responses that lowered the threshold of neutralizing antibodies required to provide durable protection. Depending on the combination of viral vectors used in the heterologous vaccination, different types of immune responses can be induced. In a study evaluating prime boost vaccination strategies for immunogenicity and protective efficacy against human malaria, researchers used 3 vaccine platforms and different prime/boost combinations, including DNA plasmids MVA vectors, and the attenuated strain Fowlpox FP9. The findings demonstrated that different vectors must be administered in specific sequential orders to induce optimal interferon gamma responses. Notably, DNA priming followed by MVA boosting and FP9 priming followed by MVA boosting proved the most immunogenic, inducing broadly specific IF and gamma responses that showed cross-reactivity against 2 Plasmodium falciparum strains. Recent years have seen the advent of viral vectors based on insect-specific viruses (ISV). Several of these viruses belong to viral families associated with animal arbovirus pathogens, such as Flaviviridae, Togaviridae and Phenuiviridae, but their replication is typically restricted to insects only. These ISVs have been engineered to produce chimeric particles vaccines expressing chimeric antigens for a range of vertebrate-infecting viruses, including flaviviruses and togaviruses. While these chimeric vaccine viruses are replication deficient in vertebrate cells, they grow to high titers in insect cells and their particles are antigenically and structurally indistinguishable from the corresponding wild-type viral pathogens. Viral vectors have existed for over 4 decades, many of which have been utilized and are currently being used as vaccines against infectious diseases. The SARS-CoV-2 pandemic has pushed forward the development of viral vector vaccines and highlighted both the strengths and limitations of some of those platforms. Further engineering of those vectors will be required to improve reactogenicity, efficacy, and vector dose. Thank you for watching. If you want to gain more knowledge, subscribe to the Creative Diagnostics channel. We are dedicated to help you succeed and advance your research progress. Visit our website for more information.